Hello, hello, hello. Welcome, everybody. Welcome, welcome, welcome. Here we are back again. Yes, this time we are so back. We are so back. I owe all of you an apology, or not quite an apology, but rather an explanation. Last night, when we were supposed to have this stream, Purkinje had an episode of what I will politely describe as antibiotic-mediated gut dysbiosis, and so that will be the official explanation for why I had to jet all of a sudden from stream last night. So first, I will begin by welcoming all of you. Hello, Silly Catboy and Archie, and hello, Marlon and John and James. And say Lalo, Flying Taco, welcome, welcome. Hello, Pamela. Hello, Blah. Hello, Otto. Welcome, welcome. Thank you for all the warm and well wishes for Purkinje. She is fighting through a lot right now in terms of battling cholangial hepatitis, but her vets believe that her prognosis is good. It's just, it is a very rocky road to recovery. So for those of you who are wondering what antibiotic-mediated gut dysbiosis means, she pooped Tox's pants last night. So that is why I had to rush out, because we were informed by our veterinarians if she had an episode like this to bring her back right away. But you know, they took her vitals and it looks like she's fine. It's just, this is what happens when you are on 3 broad spectrum antibiotics, so it is within expectation that her gut microbiome and her colon be under some duress. Hepatitis, a liver condition? Correct. So, if you don't know, the second most common liver condition a cat can get is cholangial hepatitis. So it is a GI ascending infection starting from the colon, heading up towards the liver and gallbladder. And it has not been fun for her, but we remain hopeful with regards to where her prognosis goes. She's currently chilling right now, all behavior pretty normal. So, you know, it's been rocky, but things are definitely looking brighter now than they were last night. We'll put it that way. Hello, 44 CPU, welcome. Hello, CR Smith. Proof that human neutrophils are stronger than cats. Well, the strength of human research is in fact much stronger than cat research, because of course, you know, given my propensity to read academic papers, I have been nonstop reading almost anything I could get my hands on. And let's just put it this way, if Elon Musk and or Jeff Bezos had cats and they really loved those cats, we would not have this issue of there being such a lack of research into cat science. But yes, she is doing a lot better, and I am very excited and happy to be streaming and talking to all of you tonight. But first, let's just redo that announcement real quick. For VTHEOREM, our 2 latest members, Myelin and Syzygy. You can find their information down in the video description here. Myelin is a psychologist who has been on this channel before, who helped me through a microbiome paper because I am a terrible statistician, and Myelin has been very, very helpful with regards to helping me as a dumb bio person understand statistics. This is also their eye banner, so their art is actually very, very, very good. I am I find myself sneaking peeks at this eye banner very, very frequently, just because I find the art to be very compelling. And on the bottom here is Syzygy, who is a physicist, astrophysicist, engineer, computer scientist, wearing a lot of different hats. If you like space stuff, please go check Syzygy out. Again, links in the description. Very, very exciting to be working with him in the future, so stay tuned. Alrighty. So let's begin. Let's talk about tuberculosis. Let's just get right into it. So first off here, what is tuberculosis? If you haven't watched my very first long-form video on the channel, which I I struggle to say recommend because it looks a lot different than my current style. It was, of course, modeled exactly after Tirzu, because I had no other starting point. But if you haven't seen that, tuberculosis is one of the nastiest pathogens to ever exist in all of human history, and it is responsible for a particularly nasty infection of the lungs, which is very, very difficult to clear. People who have a tuberculosis infection generally have it for life, considering that this bugger is known to hide out in the granulomas of the lung. So it is so immunoevasive that there's an equilibrium established by evolution where your immune cells will actually lock up tuberculosis in what's called a granuloma, which is like a dog pile of different immune cells, mostly macrophages. So because the immune system, or you could interpret it, I'll say this to be extremely correct, you could interpret it that the immune system has too much trouble actually clearing out a tuberculosis infection, and so this uneasy truce between tuberculosis and the immune system forms, where tuberculosis is shunted in these structures within the lungs, but they are never quite able to be eliminated because they are hiding behind this dog pile of immune cells. So in my Biochemistry 101 stream, I actually talked a bit about what makes this pathogen so frustrating and such an important part of, global research here. Do neutrophils or any non-ocular cells have any eyes, as in light-sensitive organelles? I can't think of any at the top of my head with regards to mammalian cells, but for sure there are non-mammalian cells that have ocular responding proteins, precursors to eyes, of course. So in my Biochemistry 101 stream, I wanted to make it very clear that tuberculosis, now that we are, maybe COVID is no longer the thing that is at the top of your head at all times. At this point, it's okay to think about other pathogens in terms of global importance. Tuberculosis still remains one of the greatest threats to humankind. 8 to 10 antibiotics are needed to treat multi-drug resistant tuberculosis. You can imagine that Purkinje, poor Purkinje, on 3 antibiotics is already sort of struggling to keep her insides in. And so 8 to 10 is a very unpleasant antibiotic course, and it's an antibiotic course that a lot of patients have trouble keeping, making it a very, very, very difficult bacteria to fight. Even though, throughout all of our tools of modern medicine, there's still 30% of patients experiencing treatment failure, right? So it is a very nasty pathogen, and it only continues to get nastier the longer we don't have a long-term solution for it. And as I mentioned in the Biochemistry 101 stream, the extremely drug-resistant type, so there's tuberculosis, multi-drug resistant tuberculosis, and then extremely drug-resistant tuberculosis patients, each patient costs the US government $400,000 per case. So you can imagine that this is not only an important pathogen with regards to the quality of human lives, but also in terms of just our very functioning as a society. I hope that's not too big of a doom to say outright, but I do think that if you watch John Green's series on tuberculosis, it cannot be understated how important this pathogen is to modern medicine. Hi Sang, welcome. How's the rate of pathogenic antibiotic resistance progressing? I don't know how it trends. I can only say that it would be in our best interest to make sure that it does not accelerate. Here's the thing, and a big part of what makes drug discovery for tuberculosis so difficult, mice don't get granulomas. So I mentioned earlier in stream, if you just popped in, that the reason, or part of the reason why tuberculosis is so difficult to get rid of is that it hides in these structures called granulomas, which are dog piles of immune cells that are working to box in the tuberculosis. The benefit is that you prevent tuberculosis from proliferating all over the lung, but the downside is it makes it quite hard to then access the tuberculosis. So mice are known to be tuberculosis tolerant, actually, so they don't take tuberculosis quite as hard as humans do. Part of that might be because they don't actually form this quasi-protective yet harmful structure. So our most powerful genetic tools are in mice. You could argue that you should be doing more in something closer to human, like a monkey, but a monkey is orders of magnitude more expensive. than a mouse. And you really don't want to try something crazy in a monkey because of it, right? Not only because it is cost inefficient, right? You don't want to burn $1 million, which is, I think it's in excess of $1 million to do any simian research. Not only is it expensive, but also, you know, it is in society's view of what animals deserve better treatment, something that is more human-like. You don't want to carelessly experiment on them. So mice, I repeat this over and over again. We have a culture, at least, you know, I can't speak for other countries, but the research culture in the US, at least, is very pro-animal. And I would love to do a stream where I talk about the checks and balances with regards to animal research. And I don't say this lightly, that mice have contributed immeasurably to our continued existence as a species today. But the issue is that even though mice are such powerful genetic tools in the fight against tuberculosis, they don't get granulomas. And so that is a major, major part of tuberculosis's pathophysiology that we just can't recreate in a mouse, which is really, really troubling. What is the... thank you very much, Marlon. I should not be sparkling that much when talking about tuberculosis. It is not sparkle worthy. What is different about the macrophages of mice versus humans? I don't know that answer. It's a great question, Pamela. What I can say is that mice just don't get granulomas. So the mechanism behind why that happens, not entirely sure. But it is a very good question, and one I'm sure people will be keeping in mind for the future. I'm very fascinated with the concept of phage therapies. Yeah, so we're going to be talking about whether or not phage are the secret weapon to destroy tuberculosis. Also, I don't know if I said hi to you, Clockwork, but welcome, welcome. Humanized mice, you are spoiling it. You are getting a little ahead of me. Let's pretend we didn't see that and continue down the narrative path of, oh no, mice don't get granulomas, tuberculosis is very bad. So why is tuberculosis still so molecularly stubborn? Is it similar to how macrophages clump around tattoo ink? I'm not entirely sure, but that'd be interesting to know. Oh, did I miss a question from you, CR Smith? Yeah, I did answer that. I believe there are other cells that have like opsin type proteins, especially if you look at any simpler organism than a mammal. I don't know if any other mammalian cells are light sensitive, but you can find some in the single-celled kingdoms of life. Oh, I'm sorry, I didn't see your tuberculosis question. Yeah, retype it, please. Okay, so why is tuberculosis so molecularly stubborn? I kind of teased this earlier. And part of the reason why tuberculosis is so hard to drug is because it hides in these granulomas, right? So on the left here is an actual histopathology stain of a granuloma. And here's the thing, I don't know what I'm looking at. I have no idea what this is. I don't know where the granuloma is. There's no explanation. It's very hard to find a granuloma that is labeled to the degree that would be helpful for me, so I put a cartoon next to it from this Frontiers paper over here. So we can see that in the center of the granuloma, we have proliferating tuberculosis, labeled as MTB, Mycobacterium tuberculosis, MTB, surrounded by macrophages of the apoptotic forms, so macrophages that are actively dying, as well as this foamy macrophage, which is a special type of macrophage, and then just a whole bunch of other cells. And you can imagine that if you have this many cells dogpiling around tuberculosis, preventing them from escaping, you know, it's kind of the situation of like, tuberculosis isn't like trapped in here, you know, they're trapped with me or whatever, you know, that whole meme, you know, just it's a two-way street, right? Tuberculosis can't get out, but it's also really hard to put things in. Was tuberculosis the most dangerous pathogen before smallpox elimination? Why can't we eliminate TB like we did smallpox? Excellent question. So this is a little bit of a spoiler for a future video that I'm thinking about. So smallpox is a virus, which is different than tuberculosis. So tuberculosis, being a bacteria, has a very big genetic toolkit to do all sorts of screwy things. A smallpox also responds very well to vaccination, whereas tuberculosis, I won't talk about it in this stream, so it's good that you brought it up, but tuberculosis, with their larger genetic toolkit, is capable of shifting their antigens around a lot, so making something like a vaccine strategy a lot more complicated. There's also a high amount of heterogeneity in tuberculosis, which I will talk about. So vaccines, pretty good for smallpox. For tuberculosis, a lot harder for those specific reasons. Sorry, so there's a discussion about color. Welcome, Robert. Is it the pink? The lighter pink? Yeah, the orangey bit down here, that would be the tuberculosis, and the neutrophils out here. Epithelioid macrophages. So macrophages that have been induced to form more zipper-like barriers to prevent things from escaping. Oh, you're talking about the actual histopathology. Yeah, I mean, I think it's maybe this, but I'm not entirely sure. I don't want to propagate misinformation. I'm not a doctor. Alrighty. What are the natural reservoirs of tuberculosis? I'm not entirely sure. Is there a TB vaccine at all? I believe there are some in development. I don't know if there are any that have been approved and are in use. Vaccination is, of course, the ideal strategy here, right? If you can vaccinate against tuberculosis, then that would be ideal. I do know in my tuberculosis video, I got to actually chat with some tuberculosis researchers who are taking the strategy of mass spectrometry. So basically seeing what T cells would like to detect and seeing what B cells would like to make. Because, you know, in high school, you get this very simplified image of, oh, you know, your B cell detects an antigen and makes an antibody against it. Not all antibodies are created equal. Not all antigens are created equal. You know, at the end of the day, it's still fitting puzzle pieces, and some puzzle pieces just don't like being fit. And so, you know, those researchers are aiming to see which proteins that tuberculosis makes, in the whole pool of proteins that tuberculosis makes, which one of them would be good candidates to stuff into a vaccine. So I definitely know that vaccination is a strategy that people are attempting, but for reasons I'm about to get into, it's quite difficult. Okay, so here is a little bit about why. So this is talking about drugs specifically, because I didn't really talk about vaccines, but this kind of holds true for vaccines as well. So tuberculosis has a uniquely annoying genetic resistance to getting drugged. Unlike other pathogenic bacteria, TB does not rely on horizontal gene transfer to acquire antibiotic resistance, right? So, welcome, Jim the Evo. It's widely used in many countries. America doesn't regularly use the vaccine. I wonder if that's because the vaccines for tuberculosis are not great. Because almost by definition, if they were good, then this would be less of a problem. So I am wondering. I'm not saying that that's true. I'm just hypothesizing here. Yeah. Sierra Smith. Yeah, this slide addresses that right away, right? Do you know if they do horizontal gene transfer? So they don't, is the thing, which is kind of interesting because horizontal gene transfer is a big deal with regards to pathogenic bacteria, right? So bacteria are capable of swapping plasmids. So plasmids are circular pieces of DNA. And so these circular pieces of DNA can often contain antibiotic resistance genes. So you can kind of drop ship resistance to ampicillin or moxicillin or something, and that is terrifying, right? So a bacteria that is usually small fry can just pick up genes from its environment and all of a sudden become a huge freaking deal. But actually, weirdly enough, tuberculosis doesn't do this, and that actually makes tuberculosis uniquely difficult to destroy. So why would that be? This horizontal gene transfer is a positive for many bacteria, but for tuberculosis, it actually works against its own strategy. So tuberculosis does not rely on horizontal gene transfer, which means that when it does acquire resistance, it's due to genomic mutation, right? So what does that mean? So what does it mean that the resistance is acquired genomically? Why would that be an advantage for tuberculosis, right? Why, chat, if it's genomic, why might that be an issue? It's just something to ponder. Yeah, Clockwork's got it. Each case of resistance is unique. So that means that we cannot think about tuberculosis like one homogeneous population, but rather a very heterogeneous population. So one drug just isn't going to do it. That partly explains why in multi-drug resistant tuberculosis you need 8 to 10 for an antibiotic course. Also, it's very fun to have Jim the Evo around because I am a fake microbiologist doing this mostly as a hobby. So it's awesome to hear from community members that actually know what they're talking about. Yeah, you can't treat it broadly. And maintained after replication. Yeah, so you can't lose the plasmid because there's no plasmid to care about because the evolution happens in its chromosomes. So this means that you can't just wipe out, you can't just drop a bomb on tuberculosis and wipe them all out because each individual bacterium is going to be a little different than its neighbor, meaning that tackling one quote unquote population of tuberculosis is more like tackling many. I don't know how many, but many nonetheless. Maybe because they have higher mutation rates so they could adapt faster to the immune system. I have not read anything about mutation rate, although they can do antigenic shifting, if I remember correctly. So not necessarily... I'm not going to get into those specifics because I'm not ready to talk about that in particular. Just know that even not counting mutation rate, this is still a very terrifying pathogen to deal with. I should mention my lab is all about phage therapy. Oh my goodness. I don't know. Have you read this paper, Jim? I'd be fascinated to know what you think because my thoughts on this paper are complicated. But that is very, very cool. Yeah, I'm not a phage researcher, so it'd be very helpful to have you around to sort of fill in the blanks of my knowledge. That's very, very exciting. Is that a trait unique to tuberculosis or do other genre do this too? In terms of heterogeneity, you'll see that a lot of mankind's greatest plagues have this heterogeneous phenotyping, I guess you could say. This idea that the more diverse a population is, the harder it is to deal with. That's not uncommon with regards to the stuff that we're still trying to get rid of. Does horizontal gene transfer relate to viral DNA injection? I don't know if I would use those words exactly because it's more like pseudo-bacterial mating. Bacteria can form pili, so they can kind of form a little network between them and sort of just pass off plasmids, which can be quite annoying. Does the horizontal gene resistance of tuberculosis also correspond to resisting viral DNA injection? That's a good question. I don't know too much about how phage, beyond this paper, I mean, the one paper that I read, how phage deal with tuberculosis, although that is very interesting. Why doesn't horizontal transfer also produce unique mutations? It's not... I think it's two separate ideas, right? So horizontal gene transfer is passing off an antibiotic resistance gene, a code that allows a bacteria to make something that stops the antibiotic from working. That's a little different than tuberculosis. That's just generally changing its sort of like shifting in devious ways to avoid the immune system. Okay. So enter the bacteria hunter. So I'm all of a sudden very self-conscious that Jim the Evo is in chat just because I'll just say it right now, I am hesitant in terms of my personal attitude towards phage, which is fine. Because I hear a lot of excitement, especially from my comment section about the potential of phage. And I'm not saying that they don't have potential. I'm saying that it's complicated. And I'm sure Jim the Evil will help in terms of helping me through sort of talking about the pros and cons of the phage approach. The Bacteria Hunter sounds like a good action movie name. It absolutely does. So what are bacteriophage? So bacteriophage, if you have watched my CRISPR video, you would know that these are viruses that specifically hunt and kill bacteria. They are self-regulating and propagating. So unlike a drug where you give a patient, whatever milligrams of whatever drug, and that number of milligrams gets processed by the liver and then ejected, and then you have to take more. Phage are self-regulating and propagating. So when they kill tuberculosis or they kill bacteria, they will do so, but not before they kill the bacteria. They will make copies of themselves, so they're self-propagating. So this could, you can imagine, be sort of twisted into an apocalyptic situation where like, oh, you know, this phage, you're gonna get out of control and then I'm gonna have like a zombie thing, but that's actually not going to be the case. I'm pretty confident of that, considering phage are hyper adapted to bacteria. They don't have the tools to productively infect their human hosts. They don't care. And what's also very attractive about phage therapy is the fact that they have non-overlapping mechanisms with antibiotics, right? So tuberculosis, you know, we've thrown everything in the kitchen sink against it, and it is still a persistent threat to humanity. But phage will attack tuberculosis in a way that none of those antibiotics will. That's just not possible, right? Like a lot of antibiotics involve screwing with cell wall synthesis or something like that. This is something entirely out of left field. It is replicating phage that are going to pop the bacteria from the inside. Very, very, very cool. Complicated is better than doesn't work. Exactly. And I think it's a good opportunity in this journal club to talk about phage in a way where I know that there are a lot of people excited about phage. And I want you to continue to be excited for the potential of modern medicine. But also, this is a paper that actually convinced me that it's a lot more complicated than what I initially believed as well. If I knew an antibiotic would work on an infection, I'd take that over a phage. The rub is that we can't treat with antibiotics. That's actually very in line with what I understood this paper to be conveying. That the phage might be a very powerful but specific tool that is not going to solve all of our problems, but maybe will solve a couple very important problems. We'll just say that. All right. So, phage therapy is not a new idea for tuberculosis. Oh, hey, BCG vaccinated. I totally forgot I pulled this thing out. This is actually the wrong screenshot. Unfortunate. There's a paper out there that I meant to put here for a screenshot that says phage therapy for tuberculosis, dream or delusion. Oh, I think that one's actually coming up. This is just actually showing that we've been thinking about phage therapy for TB since 1963. That's the earliest citation I could find for this idea. I don't think I have that other slide in there, but yeah, to couch the kind of drama here is that phage are not looked upon as like the what is it called, the god tool, the deus ex machina for antibacterial stuff, but that doesn't mean that they're not interesting or potentially useful. It's just I think they get a lot of hype, and we have to be careful about, we have to give phage the appropriate amount of hype. I'll put it that way. Okay. So, here it is. Yeah, mycobacteriophages to treat tuberculosis, dream or delusion. I actually, in this paper's defense, I think this paper is actually quite optimistic about the potential for phage, but there's also this like acknowledgement that the road between 1963 and 2024 with regards to phage research, as someone who has spent maybe a week studying this, has been troublesome, although I'm sure Jim can give a lot more insight in terms of what that road actually looked like. I'm merely a spectator. I'm a tourist in this area. Welcome, Engine Crafter. Welcome. So there are a couple of caveats, right? So bacteriophage are viruses that specifically hunt and kill bacteria, but they're so specific. Phage are so specific that you need to have different phage for different strains that have different receptors. So for something like tuberculosis, that's always like shifting, that's always evolving to shift away from the immune system. If it can do that to the immune system, it can definitely do that for phage. So you wouldn't be taking one phage, you would be taking a cocktail of phages. You would have to be taking so many different ones to make sure that you hit this heterogeneous population. And now I hope you're beginning to appreciate that, like, dude, we had it good with smallpox. Not like that smallpox wasn't dangerous, but the vaccine was huge, right? And hopefully this great country of mine will continue to get behind the idea that vaccines are huge. So while phage are self-regulating and propagating and kill and are great bacterial killers, you can kind of think of introducing phage like introducing predators to an ecosystem, right? Sometimes they'll outcompete and completely wreck a population, but also sometimes they might just find equilibrium, right? So in an ecological system, you can find equilibrium where there's some proportion of prey and some proportion of predator. And if you have too much prey, then predators balloon, you know, like they exponentially grow. But then when there are too many predators, the prey gets reduced. And if the prey is reduced, the predators starve. So you can have this equilibrium, at least in theory, although I'm not experienced enough to know whether or not this has been observed, you know, observed experimentally, but certainly this is an idea that can happen. Can phages get through the granuloma? Oh, we will talk about that in this paper. We had a go at smallpox. 5 initial fill 2024. Please don't tell that to any grant funding agency that I'm applying to. So not harmful to human hosts. Yeah, they're not evolved to deal with eukaryotes, and so we have very little to worry with regards to them infecting us, but because they can't infect us, they might have trouble passing through the granuloma, as Spepik is referring to. Yeah, trying to write a video about TB has been hard, so hard to focus on one thing. Yeah, I mean, I would love the opportunity to redo my TB video, just because the stories are so insane and there are so many insane stories. It's hard to just pick the interesting ones and juggle time, right? Because you can go hours and hours on TB and never get tired. Okay, so I mentioned this earlier, but mice are TB tolerant, right? So our greatest genetic tool, the mouse model, not great for tuberculosis. So it's another hurdle with regards to drug discovery. But scientists are nothing if not tenacious. Of course, it is 2024, and this is a very recent paper released in March 2024, and still we're still thinking about phage. And this is something that I think I want to speak on my soapbox about in terms of the romanticism of science, in a way where, like, I think part of what emotionally resonated with me as a graduate student was this idea that I am putting my ass out there for an idea that I believe in. And I picked a lab where they were doing very counter dogma research, I would say. So the idea that there's a functioning immune system in neurons is a counter dogmatic position. And so the fact that there are still people out there publishing in Nature, by the way, on phage therapy for tuberculosis, when the first paper was in 1963, which is not that much older than the whole history of molecular biology, by the way, it is inspiring, I think. And it'll make the story when I report to you in a couple of years that, phage were the promised solution all along. It'll make that drama feel a lot more impactful. What does humanizing mean here? Is it just to make them form granulomas? We will get into it. Very exciting technology. So let's read the title. Bacteriophage therapy for the treatment of Mycobacterium tuberculosis infections in humanized mice. So we will get into what that approach means in just a bit. So what is different this time around? So humanized mice have more human-like immune systems and therefore more human-like granulomas. So let's see how this is done. So first, you start off with a NOD mouse. So this is a mouse that is immunodeficient in innate immunity. So they have mutations in their genome that cause the maturation of macrophages and dendritic cells to stall. And so that's 2 players of the immune system already knocked out. Within these NOD mouse, you break the PRKDC gene that stops VDJ recombination from resolving. So if you looked at my recent video about the microbiome, there's a bit in there talking about how T and B cells generate the pseudo-infinite variability with which they can make things to recognize novel antigens. And they do that by scrambling their own DNA. And part of that scramble is the resolution of DNA double-stranded breaks. And so this gene helps with resolving these DNA swapping mechanics. I'm not too familiar with how this works specifically, but the machinery needed to do this recombination, that's broken when you break the PRKDC gene. Yeah. If the problem is granules in the lungs, could we just replace the lungs, like artificial lungs or a transplant? We're a little far off from being able to grow fully functional lungs for mice in a in a meaningful way. Also, that sounds very expensive. This is a lot cheaper. Still expensive, but just a lot cheaper. 3, break the IL-2R gene that hinders immune signaling and NK cell maturation. So this is another set of manipulations that is killing the immune system here. So in order to make a mouse that looks a lot more human, you first have to destroy its entire immune system, basically. And once you've done that, you take the horribly immunocompromised mice, and then you inject human stem cells, replacing the mouse's immune system with our own. Here's the problem. This NSG mouse doesn't make mature macrophages. It sounds great on paper, and I feel like every talk I've ever gone to with regards to showing off these humanized mice, this is a very hot topic. I've seen so many talks, and the people who do this work are often very excited and are very good at selling their technology. But humanized mice don't make mature macrophages in response to tuberculosis. So why is that an issue if we're studying tuberculosis? Why are macrophages a particularly important immune cell for the pathophysiology of tuberculosis? Give chat a little chance to think about this question. Can you graft a human thymus into the mice? Again, with the grafting, those are procedures that I think are unnecessarily time-consuming when actually this is a lot more efficient. Can is a different question than is practical, right? So part of why you might not see that approach is because these NSG mice do exist. Once you do the genetic engineering, you just have to pay the company to continue breeding them, and then you're off to the races. I don't hate that idea of grafting an actual organ in there, but then you have to deal with things like organ rejection, which is no good. Macrophages are where the TB makes their cute little house. Exactly. Why don't they add mature macrophages? I will gladly accept my Nobel Prize. It's a good question. I think that they have to grow up in the mice to be properly trained to not attack. Although I'm not entirely sure. That's just me hypothesizing. They introduce stem cells so that they can form into mature macrophages after the fact. Also, the stem cells come from human donors, by the way. So it is for something so artificial, that part is quite real, right? Actual human donors donated their macrophages essentially, or donated the stem cells that became macrophages for the mice. You would need macrophages to produce granulomas. Exactly. The whole thing about tuberculosis is that it makes granulomas. And so if your humanized mice doesn't make macrophages, that's going to be an issue because you are not going to capture what makes tuberculosis. So adapt and overcome. This paper improves upon the NSG mice by taking that mouse, this horribly genetically modified mutant mouse with human stem cells, this very artificial system, and they're going to make it even more artificial by adding in 3 more human immunity genes to repair the macrophage response. I harp on the fact that this is very artificial just because I think people who sell humanized mice are very good salespeople, and I believe in their data. I don't think that they're wrong. I just think that we have to remember, for the sake of our scientific humility, that this is a very artificial system, and we have to keep that in mind when we celebrate the results or decry the results, right? We just keep that in the back of your mind. All right. So here's our experimental approach. Usually I would have you, kind of figure this out, but for today, it's actually pretty simple. There's not much here. It's first test some candidate phages' ability to kill TB in a dish and / in liquid culture. So the most artificial, most simple test. Then if it passes that simple test, then go ahead and try to kill tuberculosis in macrophages. And then finally try your 2.0 humanized mice, see if tuberculosis is killed by these bacteria hunters. Have we tried breeding these mice? Is this sustainable? Could we use these guys to make packed white blood cells or B cells and antibodies? You can buy them, I think. The NSG mice, you might look, if you Google Jackson Laboratories, NSG humanized mice, you might be able to buy them. Now, I might have missed this part in immunology, but what is the mechanism behind granulomas? I can't imagine it's a part of any pre-mediated genetic response against infected macrophages. Not all lung diseases form granulomas. Oh, you would have to look very specifically about the mechanism, the different pathways with regards to what induces a macrophage to start the granuloma? I'm afraid I don't know the answer. I'm sorry, Captain. All right, so here is a fun little piece of data that we can all appreciate. Even if you have no experience in molecular biology, we are going to look at this data and interpret it, right? So we have here on the left hand here, control. And then on the left here, we have concentration of phage in, in these units, right? So control, no phage. D29 phage, CHA phage, and DS6A phage. So which one of these phage do you think deserves the most attention, we'll say? So the least concentration of phage are down here. Then you add 10 times the amount of phage, and then 100 times the amount of phage here. And you also add tuberculosis. And so if we look at the control, this is an opaque plate. That's because tuberculosis is growing all over this plate. I feel very bad for the researchers who have to work with that. And so knowing that opaqueness means high concentration of bacteria, which phage would you improve upon? D29 and D6A have visible differences. Yes, I like the way that you put it, Marlon. That's a very hypothesis-less way to describe the data, which is actually very important. And it's something that I encourage my students to do all the time, right? Before you jump to conclusions, you want to actually state your observations so that you don't bias your interpretations. So D29 and DS6A look different. That's the first thing to mentally acknowledge if you do it stepwise, because if you do it stepwise, it's a lot cleaner with regards to your thought. D29, the color change is at the lowest concentration. Exactly. So we have a clear area here where all of these tuberculosis have been killed, right? So when you increase tenfold, you get a lot more killing. There's still a patch of tuberculosis here. And then at 10 to the fourth, a very clean plate. It looks like D29 is really good at killing phage. DS6A is no slouch either. So yeah, I agree with you, chat. CHA looks unchanged. Exactly. CHA doesn't look great. In fact, a previous paper had found that CHA is, I believe they found, I could be wrong because I didn't read that paper, but I think they found that CHA infects tuberculosis but doesn't cause lysis. So this is a control for a phage that infects but doesn't kill. And we can see that this looks like the control as it should. CHA has a slight clear area at the top 100, but it could be experimental error. Yeah, it's kind of hard to tell. Yeah, but I agree. D29 and D6A look the most promising. What does that even look/feel like? It looks like some goofy, some grody putty. Agarose gel. I'm trying to think if there's any real world equivalent. Not really. I've only felt agarose. I mean, so agar is a like a jelly kind of dessert, but it's set way thicker than agar. It's a thick kind of gel. So yeah, D29 looks pretty good. In fact, if I were to choose a phage just from here, I would choose D29. However, interestingly enough, D29's ability to kill tuberculosis is not replicated in liquid culture. So here we have plates, right? So we dumped tuberculosis onto a plate that contains nutrients and agar. So tuberculosis growing on a solid medium. You do it in liquid culture, growing freely in liquid, and D29 stops working, which is bizarre, right? So if we take a look at this data, culture supernatants, OD at 600 nanometers. OD is optical density. It is a measure of the optical refraction. So imagine you have a solution of liquids, and in that liquid are suspended particles. If you shoot a beam of light through that liquid, that beam of light will scatter when it hits those particles, in which case in our case, those particles are tuberculosis. So the higher amount of scatter you have at a wavelength of 600 nanometers, the more tuberculosis is in there. It's basically saying how cloudy is the solution, if I were to put it simply. How cloudy is the solution? So when you grow liquid culture of tuberculosis, you see day 0 to day 6, they all look quite similar, but day 9, D29 here in teal squares is not different than no phage, which is here in purple. CHA, of course, still not working in liquid culture, but DS6A working just fine. So that is bizarre, and Mute is actually getting towards my next question is, why do you think that is? Why would a phage that works beautifully in plates all of a sudden not work in liquid culture? Does resting on a surface versus being in a liquid change a microbe's interaction surface? So it can, with regard to the structure of the colonies that they form, but this is a lawn. I don't know if lawns form biofilms, because I'm not a bacteria guy, but it'd be interesting to know if there are physiological differences. However, there's a different explanation for this as well. So Mute says, so D29 needs to be held together more tightly to be effective if the liquid has more flow. We're not flowing anything, we're kind of shaking the same liquid over and over again, if that makes sense. So there's not new liquid being pulled in, so the concentration should be the same. Can TB deploy a different capsid defense system in aqueous media? We're getting fancy. That's a fancy hypothesis. There's a much more simple hypothesis here. Could it be that D29 could be impeded by lysed bacteria? That's less fancy than clockwork and something that I think is interesting. Like if the TB guts are somehow inhibiting D29, that's definitely true. No, or it definitely could be true. Change of nutrients are resulting in TB developing mutations that are more efficient in cutting phage DNA. Somewhat of a fancy hypothesis as well. It's the same nutrients, actually. It's just depending on whether or not you're growing on a gel versus growing in liquid culture. Yeah. So so I'll butt in here because it's a lot less fancy than what everyone else is saying. So if we take a look here, this plate is pretty much saturated, right? There's a finite amount of space for each individual TB to grow, and these bacteria here grow, a little more slowly because if you are swimming in your food, turns out that is really great for growing. And so the thing is that when you plate the tuberculosis, you know what? Maybe I take this for granted because not everybody has worked with bacteria before, but the way that you do this is that you spread a thin solution of tuberculosis onto the plate, and wherever that tuberculosis spread is where the tuberculosis will grow, the bacteria will grow. And so in liquid culture, you don't really have that because you just keep on spinning that flask, and so you grow and you grow and you grow, and if tuberculosis dies, new tuberculosis can grow in their space, right? But here, there's an empty space here because the tuberculosis that were sitting here physically, they all died, and tuberculosis don't move on this plate. They only replicate. Does that help? So here in liquid culture, they're growing and dying and growing and dying and growing and dying. As long as there's food around, they're growing and dying. Here, the tuberculosis was put on the plate and then they sat down. And then where the blank spots are where the tuberculosis died. So there's a possibility here, a much less fancy explanation that, oh no, I didn't put the slide in here. The less fancy explanation here is that the tuberculosis, actually, I think one of you did get this as an answer. Tuberculosis developed resistance to D29 much faster. That is, I think somebody in chat actually did mention that and I misread it. Yeah, so Captain, half of your answer is maybe correct. Like developing mutations, I think, is the answer here. Where in liquid culture, because they're going to grow no matter what, because you are swimming in food, that allows tuberculosis, buys that tuberculosis time to replace the dead tuberculosis, which is why you no longer get killing in D29. D29 is maybe heavier than water, so they're actually grown by rotating the tubes. They're shaken constantly. D29 is too efficient. Don't know about that. It kills only the population of tuberculosis that it is meaning to kill. But in the plate, we would see that it has quite good general killing ability. I think developing resistance is the most likely hypothesis here. Fun conversation, though. It's interesting to see what you all come up with. Why are the error bars so big in day 9? That's just biology. I don't know what to tell you. I don't know what to tell you. All right. Okay, so DS6A, so it's not as good on plates as it is as D29 is, but overall it looks like maybe DS6A is the way to go, and Cha we can just ignore. Okay, so the next question we have is, does this phage that we have pared down in the previous experiments, do they kill TB hiding in macrophages? Why does D29 get resisted while DS6A doesn't? Here's the thing that you kind of have to be comfortable with when you do advanced biology study is sometimes you just gotta shrug your shoulders. What we can say is D29 probably the TB developed resistance much faster to D29. That's about all we can get from this data. Who knows why? That's for some other PhD to figure out. All right, so let's take a look here. So we have here the pluripotent stem cells here, and then you give them these immune factors and they become macrophages. This is how you induce macrophages. You get some stem cells, you add in some immune juice, you get macrophages, you throw on the Mycobacterium, MTB. They infect the macrophages, they love infecting macrophages, and then afterwards you add the phage. And then you check day 0, day 5, and day 10 for the presence of tuberculosis. I may have missed a bit of something. Why do we need to test both plate and tube? As you can see, it was actually a really good idea to test the plate and tube, because if you only tested the plate, you might have picked a phage that tuberculosis is very good at developing resistance to. So I don't know what the original reason for the authors to do this was. Maybe it was a grumpy reviewer who was like, you should do the plate too because blah, blah, blah. But it actually is good that they did both, because otherwise, if you just did this, you would have totally missed that it doesn't work in liquid, which is an issue. Okay, so we are evaluating whether or not tuberculosis actually kills, or the phage kills the tuberculosis in macrophages. Very exciting, very exciting. So, what are we going to find here? DS6A does in fact kill tuberculosis in macrophages, right? So if we take a look at this data, CFU, this is colony forming units. This is how many colonies are formed. Actually, that can't be right. This is in the tens of thousands, which is insane. I'm not entirely sure how they got this as like how they measured CFU in this particular experiment, because these numbers seem very large for a typical colony counting type situation. But the idea here is that DS6A, by the time you reach day 10, there's no more tuberculosis. Killed all of them by day 10, which is pretty incredible. And that is in macrophages. CFU per mil, maybe it doesn't say per mil on their axes. I didn't really read this particular data point very carefully. All right, so this is looking quite good. It's looking quite hopeful for our bacteriophage candidate DS6A, right? Cha, of course, not doing anything. In fact, it looks like D29 and Cha, actually I just noticed this. Take a look at these two phages compared to no phage. Isn't that weird? That's so weird. What am I seeing, chat? This look weird to anybody else? What the heck is going on here? I totally missed this at first. Can someone tell me why my brain is exploding right now? That's actually really weird, yeah. Hormesis? I'm not exactly sure what that means. But why are those first 6 bars so surprising? More TB in day 10 than no phage? Yeah. What? What? Why is there more tuberculosis when there are phage? That is actually that's really bizarre. And I don't have an answer to that. Another interesting point of data in this paper. Okay, so we know that D6A kills tuberculosis in macrophages. But is there an alternate hypothesis out there? This data is only measuring the load of tuberculosis. So what is a potential alternate hypothesis that is happening here? Hormesis is a biological principle that describes how organisms and cells can adapt to moderate stress to improve their functionality and tolerance to more severe challenges. Yeah, interesting. Yeah, I mean, maybe. TB activated defense mode invited more. Maybe. Maybe. I mean, it certainly looks plausible. Maybe phages also affect the macrophages. Yeah. You know, it makes a lot of sense. You know, if we killed all the tuberculosis, did we also just kill all the macrophages? Yeah, that's great. Yeah, good job. That is exactly the control that I would like here. And of course, we do have that control here. This is immunofluorescent data, so DAPI. This is a stain that binds to DNA. So what you are seeing in blue here are the nuclei of these macrophages. In here, in green, we have GFP. So this is infection with tuberculosis that has been genetically rigged to make green fluorescence protein, so that if we see green, we know which cells are infected. Cell mask, this is something I've never actually used before, but this is apparently a membrane dye, so you can see the shape of the macrophages. And then the merge. This is very hard to see, so I actually took this image and I photoshopped it to make it brighter. It's still not great, but hopefully you can see what we are looking at, right? So uninfected, no tuberculosis. We have our macrophage nuclei up here, no tuberculosis here, so no green. These are happy looking macrophages. You add tuberculosis, then you get some infection, right? There's a green dot here, green dot there, green dot there, green dot there, green dot there. So you get successful infection. Then when you add the phage and the tuberculosis, you don't get any of that green here, indicating that this phage was very efficient at killing the tuberculosis residing in these macrophages, and that these macrophages are still intact, right? This is not a cell viability stain, so I have no idea if these macrophages are actually alive, but I'm not too fussed about that. They don't look like they've blown up. They look morphologically, they look the same as the rest, so I'll take that. I think that's fine. Okay, I mean, yeah, so far so good, right, chat? Like, this D6A, that it kills TB in a dish and in liquid culture, and it kills tuberculosis in macrophages. So far so good, very exciting, very cool. But now comes the actual moment of terror, right? The ability to kill tuberculosis in an actual animal is a completely different ballgame than doing it in a dish in various different ways. This looks pretty definitive so far. So far, ... exactly. Those ... are very, very important. Is it practical to deactivate the ability of TB to survive in a lysosome? You would have to have a very specific way to do that, which we can hypothesize at the end. I would actually love to spend the chatting portion of stream just dreaming of ways we could fight tuberculosis. That'd be very fun, actually. Okay, so what about its ability to kill tuberculosis in an actual living, breathing, honest-to-God animal? First, we have to validate that our humanized mice 2.0 actually has a good chance of working. And so they characterized the immune cell population of the humanized mice before and after infection to see whether or not we get macrophages activated. So we have a pie chart that shows the relative proportion of immune markers, which are proxies for the population of these cells, right? So CD8 and CD4, these are immune markers for T cells. So which two slices? It's this slice and this slice. These are our T cells. CD19, these are our B cells, the antibody cells. T cells are the assassins of the immune system. B cells are the antibody producing long-term defense of the immune system. NK cells in yellow, you can see that they're hardly there. NK cells are, I call them like spec ops or something, like very highly specialized killers of viruses and cancer. And then here in this teal looking blue, macrophages. So before infection, and then after infection, we get a huge, huge shift to a macrophage response and a CD8 T cell response. So very cool. It seems like, macrophages are involved. But why don't I love this as a way to show this data? What's bothering me about this data? The argument that the authors are trying to make is that the humanized mice are in fact humanized, and they are going to be good because they have a shift towards a macrophage immune response. And if we have a shift towards the macrophage immune response, then we are likely going to get granulomas, fingers crossed. But why don't I love a pie chart here? Yeah, no scale, no numbers, doesn't show absolute changes, no total cell count, exactly, where numbers? Yes, exactly. They do have like cell sorting data, but I would really love a bar graph instead of a pie chart, right? I would like to see, before infection, this absolute number of macrophage marker, and then after infection, this absolute number of macrophage marker, just so that this pie chart isn't actually saying, oh, actually B cells all died, and that's why by proportion macrophages are a lot more important, right? Like, I don't love it, but I grit my teeth and I move on. Mute is right. Where's the control mouse? So why do we need a control mouse? So I love the idea of asking for as many controls as possible, but again, in terms of like the culture that we have here of like not using animals carelessly, I need to know why you think we should have a no infection, because before infection, this is our control mouse, right? So why would we need an additional control mouse? No comparison to human responses. That's okay, because this is not a human paper. So if you wanted to, you could certainly look up a human paper and compare. So I'm not going to fault them for that. Yeah, what happened to B cells? I have no idea. This is crazy, right? Like, there's so many unanswered questions. B cells just gave up. I don't actually know if tuberculosis has specific ways to assassinate B cells or do that kind of thing. My tuberculosis video was so long ago, I don't remember anything specific from back then. I'm too naive to understand what this slide is showing. So this is the proportion of different immune cells, or at least immune markers. So we have mostly B cells, B cell markers here, followed by T cells. And then after tuberculosis infection, we've moved to macrophages and then this other type of T cell. And it's important because we want a large macrophage response from our artificial mouse, because the normal mouse doesn't make granulomas, which is a macrophage response. No error bars? Yeah, I'm not too picky about error bars here. Mostly I want bar graphs to show me after infection there's a 200% increase in macrophage response. That would make me a lot more comfortable than macrophages are a larger slice of the pie, because the pie is always 100%. So imagine that instead of having more macrophages, you actually just have fewer B cells, and that's why this looks bigger. It's kind of tricky. No absolute value, just relative. Exactly. That's why the pie chart I don't really love. I was always taught in stats to include error bars. Yeah, this is more like a qualitative thing. I'm not too like bothered by lack of error bar in a pie chart. Okay, so here is actual data that does concern me. So this is just like, for me, a little annoying. This is actually concerning me. So you infect these humanized mice, and then you check to see whether or not the tuberculosis takes hold in the mouse. This is colony forming units, so this is counting bacteria per milliliter of homogenate of the lung and the spleen. So why is this sus? Why is this sus looking at this data? Humanized mice 2.0 have extrapulmonary tuberculosis infection. And why does that bother me? Error bars overlap? That's not exactly my concern here. It's okay if error bars overlap. They do all the time in science. And sometimes, even if the error bars overlap, you can still get significance depending on the spread of your data. Why are they in the spleen, says Otto. Is that normal? Yeah, why are they in the spleen? Tuberculosis is an infection of the lung. So why are they in the spleen? I didn't realize TB took up residence in the spleen. There's good reason for that. It's very unusual here. TB in humans don't form granules in the spleen, so generalizability might be low. I don't really care about the granuloma in the spleen. I'm concerned that they're in the spleen to begin with. A lot more on Otto's wavelength. Does that mean the spleen was infected, or was it just that it was just there? So the spleen, you know, given, I forget exactly how they got the TB in the mouse. I think it was injection, intraperitoneal injection. And so intraperitoneal or intravenous, I don't remember. But the point is that they injected it somewhere, and now we find tuberculosis in the spleen. And this is not like, I don't need a non-infected control here to know that this is crazy, because CFU per mil, we are in the 10 to the 7 area. Right? So a mouse that has no tuberculosis is going to be at 0. So yeah, tuberculosis in the spleen. Very, very bizarre. TB, can TB cause a generalized blood infection? Bacteremia, I think it's called. So here's the thing. extrapulmonary TB is not common in humans. It happens, as I Googled, right? Because I was shocked. I was like, what the heck is tuberculosis doing in the spleen of all places? So you can get a splenic infection in humans, but I think, and I could be getting this wrong, so take it with a grain of salt, I think only 10% of TB infections in humans end up in the spleen. So why is that a little tricky? Why is that a little sus? Just a little sus. I don't think it's tremendously sus, but it is a little sus. Why did they decide to look at the spleen specifically here? I think they wanted to check. I think what they were hoping for is that it wouldn't be in the spleen. I think they were hoping and praying against all odds that they wouldn't find their bacteria in the spleen. The spleen is a site where blood gets cleaned, and so if you have circulation of something in the bloodstream, it is going to be, it's going to end up in the spleen. But I think they were hoping that not a lot of it was going to end up there, because as you can maybe predict, this is going to get messy now. Does that imply there's an issue / difference with the immune system function? I would be even more general than that. I would say that this, being generous, does not recapitulate the biology of TB infection in 90% of humans, which is an issue for me. This is maybe applicable if you have extrapulmonary tuberculosis, so I don't think this data is useless, but when I saw this, I was like, okay, so I have to now take everything with a grain of salt, because whatever is coming next, we have to remember that TB is also in the spleen. So that's an issue. And who knows where else it is, by the way. They checked the spleen. I don't remember if they've checked elsewhere, but we know that whatever is coming next can no longer be explained by tuberculosis in the lung alone. Do mice normally get infected with TB in the spleen? They are different animals and are common ancestors quite far back. It's a fair question, but we care about the lung in particular, and if we're comparing to human, we don't want to see it in the spleen. Yeah. The spleen stores a lot of blood. A major component of blood is macrophages. Might explain it, but that doesn't explain why the TB set up shop there specifically. Yeah, I think it's just because they inject it through the bloodstream, and the spleen will see it if it is in the blood. Is this data from a single mouse or the whole cohort? So each individual one of these dots is a different mouse. Exposure method may have been too different. Yeah, maybe if they had chosen to do it through, you know, an aerosol or something, this would be different. So that's why I think someone else had too specific of a hypothesis. Oh yeah, Myelin was a little specific. Maybe something's working substantially altered way in the human mice, just like something is different, right? And then someone else also had a specific hypothesis. I would not actually adopt any one of these in that much detail, although that's a good place to start with regards to thinking about where to go from here. Not saying, Myelin, that you're wrong, but like in terms of thinking in the cleanest way, something is different, and that's all we can say here. It should not be in the spleen if we wanted to recapitulate human biology. Although it's not useless, it's just like we gotta slow our roll here. We gotta pump the brakes and go slow. These mice have weird Frankenstein immune systems. Yeah, it could definitely be that like they have a weird immune system, or it could be the injection, or you know, it could be any number of things. Maybe they should have aerosolized it. Who knows? It's probably safer for the researchers to inject, you know, because aerosolizing tuberculosis, kind of don't want to be in a lab that does that. So I can't blame them. And this is the tragedy and the nuance of science, right? Like, this is a result where I'm not angry, you know, I'm not mad at them. I'm actually quite sad for them, right? Because now things are going to get hard. Now things are going to get complicated. Why is the sample size only 9? Is that enough to draw any real conclusions? Here's the thing that you have to be very comfortable with regards to animal research. You don't want high sample size. I hate when I go on Reddit and there are people crapping on research because it's done in a handful of mice. Look, I worked with mice. I'm not mad at you, CR Smith. I'm remembering these sort of, you know, it's not their fault either. It's just maybe a little bit of ignorance, but I treated my mice as well as I could have treated them, right? Like, I have my own pet, right? I have Purkinje. I did not like the idea of animal lives being used for research, and so low ends are actually acceptable. And so you just have to trust that the researchers did power analysis correctly. And also in this particular result, you don't need a power analysis or even a statistical comparison, because we don't care that we don't care about the difference between spleen and lung. We care that spleen is greater than 0. So we want to always minimize the amount of mice that we use. And to do that, we do a power analysis to inform the minimum number of mice that we would need for a given amount of statistical power. So for something like a Nature paper, I will usually just trust that they did their statistics correctly because it is a more high profile journal. Yeah, so to just like soapbox a bit, because it is like the eternal bane of my existence being a biologist and having to talk so often about mice that are undergoing terrible things. I think if it makes you feel better, or it certainly makes me feel better, but I don't think any mice died during this experiment due to infection, because I think, again, mice are somewhat tolerant of tuberculosis. In mice, TB dissemination to the spleen is common. Thank you, Jim the Evo. Thank you for clearing that up. It is not common in humans, from what I understand. So I guess this is maybe within their expectation, but it still, I think, dirties the water a little bit with regards to what is coming up, though. Okay. All right, so let's talk about whether or not humanized mice benefit from mycobacteriophage treatment. So here is their setup. So we have irradiation destroying the mouse's immune system, inject the human stem cells through the tail, 10 to 15 weeks, so that a human immune system can colonize the mouse. Then you make sure that the human cells have taken root in the mouse. You infect with tuberculosis, you start phage treatment, and then you check to see whether or not the lung function improved, and then check the lung, spleen, and blood. All right, so that is how we are going to be testing the efficacy of mycobacteriophage for the treatment of tuberculosis. All right. So, mycobacteriophage treated mice have a healthier weight than untreated mice. Or actually, if I were being really careful, what I would say is that mycobacteriophage treated mice have a higher weight than untreated mice. So we don't have any mice that were not treated with tuberculosis, which to me is kind of annoying. Like, I know I made the grand speech about like, oh, we should minimize the number of mice used in experiments, but like just measuring a mouse's normal weight over this time course is like, you know, oh wait, this is body weight change of initial weight %. Yeah, then you don't need an untreated mouse, because an untreated mouse over this course of time is not going to gain that much weight regularly. What age did they do this in? Sorry. How old were these mice? I wish I knew. I wish I thought about thinking, because like mice reach sort of sexual maturity after 2 months, and then I don't remember when they plateau in weight. They can live up to 2 years. So I'm just kind of wondering if having a non-infected mouse would have actually made a difference here. Maybe not. Anyway, what is clear is that on day 20 and day 22, that the phage-treated mice have a higher weight compared to the tuberculosis no-phage mice. And so we use weight as sort of a general proxy for mouse health. So it looks like this is a promising start, although it does take a lot of time. I mean, my goodness, 20 days out for you to see an improvement in weight, which is kind of shocking to me. Yeah. Gamer mice might gain weight over that much time. For sure. Some mice are munchers. They can get quite tubby. I saw a cool picture of a monument to the mice sacrificed to science. Yeah, I think that that statue is in Russia or something. I do feel quite a lot for mice that are used in research, and I have to keep on reminding myself that like their quality of life in a cage, not the worst. They have enrichment. If you give them enrichment, they have ad libitum food. You know, they just eat however much they want. They sleep. They have their brothers and sisters around. And you can tell if they're stressed. If they're stressed, they'll overgroom, they'll fight. So I have to tell myself, because I wouldn't have even classified myself as an animal lover until I started working with them. But I have to tell myself that like we would know if the mouse is miserable, and we can do things to fix it. So I don't know. What's my opinion on flies used for research? We will talk about flies ad nauseum with regards to cell and developmental biology, which I can't wait to get into in my 101 series. Okay, so gaining weight, that's good. That is a proxy for health, but now let's talk about the lung, right? So here we have 3 different metrics for the functioning of the lung. We have what is called compliance, elasticity, and resistance. And so in purple, we have the phage, and in the purple, we have no phage, in blue, we have phage. So it looks like there's not a huge change in compliance with regards to either treatment group. So compliance is, and people in the medical community can correct me if I'm wrong because I looked this up shortly before stream, but I believe compliance is a metric of how easy is it to fill the lung with stuff. So that is slightly different than elasticity, which is a measure of how easy it is for the lung to return to its relaxed state, which is different than resistance, which is how hard do you have to work to stretch the lung. I'm sure there are physiologists out there who care a lot about this, but for me, I'm just like, okay, this is lung stuff, so let's take a look. So no difference in how easy it is to fill the lung, sure. And it looks like some marked improvement, I think, in terms of how easy it is for the lung to return to relaxed. And then it is a lot more or a lot less resistant to change compared to the tuberculosis mice. Lung volume, it is not statistically significant, but it does look like it is trending towards larger lungs, although it is hard to say. So it looks like there are some improvements in the lung after phage treatment, which is really, really promising, right? Very, very promising. Is there a control missing here? So I will ask for an additional control here, and it's actually a control that someone in chat had mentioned before. A very important control, I think, that would be better suited in this experiment. Because as someone who is not a physiologist, I don't really know how good this phage is working. No TB is missing. Exactly right, Hair Day. I would love to see a 3rd bar of just normal mice. Or rather, the humanized mice that have not been infected with tuberculosis, right? Because to me, this looks like a big difference, but if I look at the number, this is like 6 down to 2. Is that a big deal? I don't know. So I would have liked to see a healthy mouse to see if this is like, oh, you know, we celebrate, the phage is doing so good, or if this is like, the phage kind of helps, but, you know, like, whatever. This is actually where I would advocate for the use of more mice. Mice plus phage minus TB, exactly. That would be useful too, actually, to see if the phage have any inherent effect. But I think, and again, correct me if I'm wrong, I think we kind of assume that phage are not harmful to us just because, again, they are so hyper evolved for bacteria that it's almost a given, or it's almost a waste of animal life. You know, that is an assumption that I am making and not something that I hold with a lot of conviction. We love uninfected control groups. Yeah, exactly. I would love one here. I don't always need one, but here I would love one. Surely they did these lung function tests on the mice before they did the experiment, right? Surely some other group has done it, but in terms of these particular NSG humanized 2.0 mice, I don't know if anyone else has used them to do this particular test. This graph could technically indicate that TB isn't harmful and phages get super abilities. Yeah, exactly. Without a non-infected control, like you've all been saying, it's kind of hard to tell. Yeah, and Pamela, you are getting to another control with his plus phage, but I'm okay with not sacrificing an animal life for the plus phage because I don't think we'll see anything. That's like, you can't use that logic every single time, right? But I think, again, could be wrong, I think they're generally pretty safe, which is why we don't bother. Okay. Here is the most shocking figure maybe that I have ever covered on this channel. Look at this figure, chat, and I just want you to take what you are seeing in. I just went over, you know, all this phage. It kills in macrophages, it kills in addition and liquid culture, and, you know, it has all this promise. And the mice, you know, they have a healthier body weight and their lungs improve. but there aren't any less tuberculosis in the lung. P equals 0.0565. Sorry. Sorry about that. Try again. That is devastating. But they're healthier. Exactly right, Spepik. What about the spleen? Engine Crafter, you are on the right track here in terms of where my brain went to. It's not a stupid question. It's probably the most important question that I have for this section of the paper. Same amount of TB, but less lung damage, maybe? How? Why? It's so confusing. Why would that be? P thresholds are arbitrary. I know. P thresholds are arbitrary. Sometimes it's significant, sometimes it's not. But it's still... The fact of the matter here is that there are phage-treated mice. If you look at the individual spread, right? Some mice do see a dramatic decrease in CFU per mil in the lung. Some mice, not at all. And that's what's shocking to me. That this thing that works so well in a dish, the difference is not huge. And it's annoying that this is a log, a log plot, right? This goes up by 10, that's like pH. And so this absolute difference is large, absolutely, but in terms of like a non-infected mouse, it should be 0, right? If we eliminated the infection. And the initial infection is down here at 100, right? So we're not even down to that level yet. So some mice, it appears that the phage maybe brought down tuberculosis, but in a lot of other of those mice, it's within the same range as the control, just the tuberculosis. That's very bizarre. Are these immunocompromised mice? These are humanized mice with human immune systems. Correct me if I'm wrong, but is that a log? It is a log. What else is the immune system doing? Yeah, I don't know. And we're relying on the phage to kill these tuberculosis. That's what we're testing. So, Engine Crafter's got a great follow-up question. What about the spleen? Oh, it's great in the spleen. It's amazing in the spleen. This is insane. Look at this. 10,000 CFU per mil in the spleen in the Microbacterium, reduced tenfold. And so many of these, look, down here, down at the bottom, so many of these mice almost have a completely clean spleen, no tuberculosis left over. Bottom of the bar graph. And that's insane. I mean, this spread is also insane, right? Bimodal. For some, it doesn't look like it worked at all. For others, completely gone. So, we are juggling this result, cleared in the spleen, with this result, which is really tough because maybe it worked for some mice, maybe it didn't work for others. The data are clearly shows bimodal. Exactly, it is very bimodal here. Huge cluster down here where it worked fantastically. The crazy thing, though, is that if we take a scan, I think this is a CT scan, but I'm not 100% sure. I'm not a doctor, but basically opaque is like damaged tissue here, or like inflamed tissue. And on the here we have tuberculosis and then phage treated. We can see, especially down here, there's a lot more damaged tissue in the lung here than there is in the phage treated. So it's working in the lung maybe, but like it's not disappearing from the lung. So like, what the hell? What the heck is this? This is why this paper, I think, drives me crazy. And I think like, I really feel for the researchers, like when they got this, it's an unfortunate p-value, absolutely. And I know that p-values are arbitrary, but still, like, if we want to be strict about our interpretation, when the researchers found this, I bet their stomachs dropped, right? Because now this is a complicated story. Okay, hypothesis time, right? Oh, a CAT scan, maybe. I'm not entirely sure. Hypothesis time, right? It's free space. So mycobacteriophage treatment does not reduce, if we are being strict about P of 0.05, if we're being strict about that, and I know it's controversial, if we are being strict about that, we can at least say that, the effect is not enormous for the average mouse in that sample. Mycobacteriophage treatment does not reduce tuberculosis bacterial load in the lung, although it trends that way. Yet treatment with the phage does improve lung function. So I will take your hypotheses. What is going on here? What is going on here? Mouse scan, true. I guess it kind of makes sense as well. Spleen is more of a liquid environment, right? Although that's not a very scientific reason. I think that there is an easier explanation than that. One that I think is more supported by the current data. Put it that way. Does phage induce a less virulent mode in TB? It's so busy not dying, it isn't damaging the body. Ooh, Otto, that's a great hypothesis. That's actually not one of the ones that I even came up with. I'll say that that is very cool. And I don't know the answer to that, but that is, if I had the wherewithal to edit my PowerPoint, I would definitely add that, because that's a great hypothesis. Smaller globs so the lungs are hurt less. I'm not entirely sure what you mean by that. Can we observe the mice's lungs to compare them with that form granulomas? I don't think they did that in this paper in terms of physiological inspection of the lung. I think maybe they did. Surely they must have shown that they form granulomas at some point. I don't think I have that figure in here, but surely let's assume they do. Let's assume that this is the perfect humanized mice. Although you are right, I don't remember seeing a picture of a granuloma in the paper, although somebody might want to pull that up for me. The phage might have taken out some of the other bacteria to reducing the overall bacterial load. The thing is that in the lung, it's not that different from most of the mice. Although the spleen, it seems to be incredible. Also, welcome, Myelin. I don't think I welcomed you into the stream. You should go follow Myelin. Myelin, I shouted you out at the beginning of the stream. New member of VTHEOREM doing psychology and the psychology of all of your favorite fandoms, which is very, very fun. Can it be because TB resistance is not horizontal gene transfer, but mutagenic? You're gonna have to elaborate that and make the connection clearer for me, because I'm not entirely sure where you're going with that line of thought. How were the phages introduced? Did they just inject to the blood? They were injected. They were injected, I believe. Same question as Ryan. How are the phages administered? Injection. That is why they actually end up in the spleen, probably because all roads lead to Rome, right? All blood leads to the spleen. Same idea. If it changes the amount of TB in the blood, wouldn't that change the effectiveness of the lung as well? That is one of the hypotheses that I have on my next slide. Very good, Spepix. So it could be that cleaning the spleen of tuberculosis improves general health to the point where you do get improvement of lung function just because you have better blood. I'm not a doctor. I don't know if that's plausible, but in terms of the data I see, that's certainly one of the hypotheses. Assume a perfect humanized mouth in a spherical frictionless environment. True. Jealous of physicists that they get to do that. Earlier, we said that TB cannot be treated broadly, so can there be a sample of TB that responds well to phages and a sample of TB that does not? So, in the spleen, it looks like all the TB were eliminated, and this is the same TB that exists in the lung, but the lung, it doesn't really work there. So that's really bizarre. Spherical humanized mouth with pi equal 3. Yeah, with pi equal 3. True. Yeah, so I really like Otto's hypothesis, and I also approve, or I am in line with Spepic's hypothesis as well, that maybe fixing the spleen explains all of this, because it's certainly not making a huge difference in the tuberculosis load in the lung. Maybe the spleen filtered out the phages. Yeah, I mean, circulating phages at least. Yeah. I thought the spleen showed it was half eliminated and the other half was only stunted a little bit. Yeah, that is true. That is true. Some of them completely gone, others not really that different. But still, the highest load in the phage treated group is still only higher than like 1 in the Mycobacterium group. Yeah, so here are my hypotheses here, right? First hypothesis, mycobacteriophage may be clearing out a systemic infection that might just improve overall health. Remember, we only measured the spleen. what about the rest of the body, right? How extrapulmonary are we talking here with these mice? Maybe adding the phage cleared the TB infection everywhere else except the lung, and that's why those mice are feeling better. That sucks. That really sucks in terms of like being able to celebrate a big W. And I kind of wish, Jim, that Jim, you are here. I was, oh my God, that's so funny. I was just like, I would like your opinion because you seem to know, I mean, you're a phage expert, but if you happen to know how extrapulmonary TB infections are in mice, because I don't know if you were here for the data that I showed, but in the lung, it didn't look like that phage did a whole bunch of anything with regards to TB load. But in the spleen, it almost cleared up, for a couple of the mice, it almost cleaned them up entirely. So we do get increased lung function, but we don't really get a clearing of the TB from the lung. And I think one hypothesis is that if extrapulmonary tuberculosis infection in mice is very extrapulmonary and not just in the spleen, that getting rid of the phage everywhere else could just lead to better health. I don't know what you think about that, Jim. I'd be really, really curious to hear your opinion on that. What is the immune response to these phages? Is it causing the same immune cascade? Maybe they're attacking the phage and not the TB as much. Oh, C.R. Smith, I really like that. And in fact, I, for time, did not include that in this. It is in the paper. They tested whether or not the human body was making antibodies against the phage instead of the tuberculosis. And you could open the paper in the description. It's like the last figure or something, and that'll actually give you your answer. Great question. I was a little lazy and I didn't include it. But that's awesome that you thought about that. That's a really fun hypothesis. And I'll read Jim's comment because it looks interesting. If you are interested more along the idea of phage changing pathogens, phage steering, conflict of interest. I coined the term. Ooh, big shot over here. Maybe I'll do maybe I'll do a phage. Maybe I'll do a phage steering journal club. Oh, that would be so fun. Jim, you up for a collab? Conflict of interest. But I would be very down to collab if you ever want to talk about phage steering on stream. That'd be very cool. Okay, so maybe it's not clearing out a systemic infection. Maybe we're still thinking about the lung, right? So maybe TB outside the granuloma is the issue, and maybe the phage is keeping TB locked up in the granuloma. Like maybe like what's really important is the TB that the TB that escapes. So maybe it's still doing something in the lung, but it's not really getting into the granulomas or something. That's certainly a hypothesis. And then maybe there aren't enough ends to see a real difference, right? Patient heterogeneity, phage penetrance. As I said earlier, TB is highly, highly heterogeneous, which means that some phage, if you just inject one species of phage, it might not be able to hit every population of TB. So maybe we just needed more animals, more tests to see an actual difference. But even so, I don't think this is okay. The paper is a great start, but I think this figure is maybe more on the depressing side for me personally. That could be an issue with phages. Host makes antibody against the phage. Absolutely. And for Pamela, the end of the paper, if you open the paper, and it is open access, so you can just scroll all the way to the bottom. Or at the end, when we do our little chatting session, maybe I will pull that figure up myself and we can just take a look at it together to see the answer to that if the host is making antibodies against the phage. I'm no biology expert. To be honest, I'm a liberal arts major, mostly in history. That's very cool. Glad I got something from my few biology classes. I think biology is for everybody. I think there are some sciences that are very complicated and require a lot of mathematical know-how, but biology, it's just little guys doing little things, and I think anybody can get behind that. But I think that's not to downplay your achievement, C.R. Smith. It is to highlight the fact that with a little bit of work, this is for everybody. And I am glad that you have put in the work to have caught me in a moment of weakness, my moment of sweeping something under the rug because I didn't want to talk about it. but that's genuinely very cool that you were able to come up with that. Because the spleen effect was so bimodal, I would really like to see the statistical significance between the distribution of the not entirely eliminated CFU per mil and the untreated CFU per mil. Yeah, I'm not like a stats head, so I don't have anything additional to add to that, but I do think that that would be interesting. Okay. So, this slide is basically pointing out that we have a heterogeneous population, right? Tuberculosis, it is pretty good at colonizing the lung. It's very tight up here in this ridiculous CFU per mil figure, but phage, maybe it's because the immune systems of the mice are different enough where you have this different result, this heterogeneity, this high variance, or it's because the population of phage, the population of tuberculosis that colonized this mouse that was very vulnerable to the phage is maybe different than the colony that made it up here. That's one hypothesis so far, although we will see whether or not that hypothesis holds water in the future. Okay. Yeah, I do wonder how this correlates with lung function. So is this mouse the best lung performer? I would have really liked to see a correlation, you know, a line plot between these two functions. Looking through the papers, it never explicitly states that the mice used formed granulomas. Oh, that's kind of an issue. I just kind of gave them the credit. I was being very unjudgmental, but Spepik, that might be a freaking issue if your mice are not forming granulomas. I just assumed that they did, and so the rest of this presentation will assume that they did, but you are right on the money. It would have been nice to see whether or not they made granulomas. I have a lot of knowledge about biology, but I'm really interested in learning more. Yeah, biology's awesome, as you can tell. So one hypothesis is that we have some phage resistance going on, and that's why we don't get full penetrance into the lung. Now remember, if these tuberculosis are forming granulomas, that gives them the opportunity to filter out or block a lot of the incoming drugs. And so maybe the reason why we don't get effective clearance in the lung is that they've adapted phage resistance. So they actually did this kind of neat experiment where they added tuberculosis from the mice, and then they added some different phage. And so if you add tuberculosis isolated from the lung and spleen, and then you add your normal control phage that doesn't kill, you get a bacterial colony. If you add a control phage and you add the tuberculosis that were treated with the killing phage, you get very little growth. Killing phage plus tuberculosis, you get no phage, right? The killing phage killed all the tuberculosis. And then this one tests for resistance, right? So you take your killing phage, and then you take your phage you take your tuberculosis that were already treated with phage. So you still get nothing, which means phage resistance was not acquired by the treatment group. Yeah. We need therapies that inhibit or reverse granuloma formation. This will help with phage and antibiotic treatments. That would certainly be wonderful. I would not be brave enough to try that. Because my cell biology is not super duper strong with regards to understanding the pathophysiology of granulomas, but it seems that granulomas are kind of an architectural problem, and part of that issue is that not all the cells in a granuloma are alive, and if the cell is dead, it can't respond to the stuff that we do to it. So it's kind of interesting. If no phage resistance, no phage steering. Also, why did the control phage now kill? Oh, sorry. So this is using the control phage on the tuberculosis that were already treated with the DS6A phage. So this is like a double treatment. And so there's a little bit of growth just because this DS6A phage killed most of the tuberculosis. Yeah. Okay. So location, right? The granuloma, this thing from hell that's making tuberculosis so hard to fight. You can imagine that one of the reasons why we don't get phage killing is maybe it's just being stuffed out of the granuloma, right? The granuloma is a tight structure full of these epithelioid macrophages that have zipper-like membranes that form a tight barrier, preventing things from leaving and things from getting in, which has been a huge headache for delivery of drugs. And so you can imagine that maybe there are some phage that sneak in and can do a little bit of killing, but then the TB rapidly acclimates, and then maybe some of these other phage just get blocked by these membranes. Kind of annoying that way. Yeah. So here are my conclusions. Mycobacteriophage D6SA has the potential to be an anti-tubercular agent. I think that is true. Here's the thing. What I can say, what I can't take away from the authors is that you add this phage to mice that are infected with tuberculosis and have high bacterial load, they get better. That's not nothing. That is optimistic. That has potential. It has shown efficacy in killing TB in vitro and in macrophages, right? So in a dish, in a tube, it does very, very well. And in a tube with macrophages, it also does very, very well. And it shows efficacy in clearing extrapulmonary TB infections. So at the very least, maybe this is something for people who have very bad systemic TB infection and can help them clear the infection out of places like the spleen. And this is not a total loss, right? Like, I'm bummed out by the fact that it didn't decrease bacterial load in the lung, but the battle doesn't end here, right? There's a reason I think that this is in nature, although I think the reason why it's in nature is because it has potential. I think on its own, it's not super flashy. They use a very interesting novel mouse, but I think the end result leaves you wanting for more. It's like having half a bag of potato chips, right? Like, the chips were tasting good and then someone snatched the bag away from you and now you're left with this craving. It's not the worst thing in the world. I think the researchers were probably sad that they didn't see a lot more of that tuberculosis get cleared from the lung. But, you know, this is research, and research marches forward, right? This is a duplicate slide. So what research could that lead to, right? So we know that this phage, if it can get to the tuberculosis, is really good at killing it. What I think is happening is that it just can't reach the TB. And part of the support for the idea that maybe TB are forming granulomas in these mice is the fact that the phage can't kill them, right? Maybe it's because they can't circulate to the lung. Maybe this would have been better as an aerosol. Maybe that's definitely true. But one potential hypothesis is that you do get successful formation of granulomas, and that's what's making this drug delivery so difficult. So the battle doesn't end here, right? There are a lot of other tricks that can be done to improve the penetrance of the phage to the site of infection. So there are these molecules called cell-penetrating peptides. These are proteins that can help molecules sneak through cell membranes. So HIV TAT. So this is the first cell-penetrating peptide that was discovered. This is the TAT protein from the HIV virus, which I hope you are all very well familiar with by this point. Not that I've covered it on my channel, but I just remember learning about it nonstop when I was growing up. And so TAT is a protein that helps HIV get into the cell, and we've actually co-opted that for bioengineering purposes. So maybe one possibility is to engineer this phage by sticking a cell-penetrating peptide on it to help it pass through these membranes. Maybe it was the injection method, right? Maybe injecting phage is not the smartest idea because it all ends up in the spleen. Maybe there's a way you can develop an aerosol that helps deliver it directly to the lung and no other places. Maybe you could stick it in a liposome, right? A little fatty cell that can dissolve into a membrane. Maybe that's an approach. Antibody-mediated endocytosis, stick an antibody that's specific for something in the granuloma and have it traffic that way. Maybe gold lipid nanoparticles. These are very, very hot right now in drug design in terms of finding technologies that can help get drugs to the locations there need to be. So even though I was a little doom and gloom this paper with regards to like, it's a bummer. I mean, here's the thing, for Jim the Evo out there who's a phage researcher, the phage did a wonderful job in the spleen, and it probably did a wonderful job the rest of the body too. Phage alone is not going to solve the TB solution. I think maybe phage and a combination of antibiotics and a combination of very talented engineers to get them into a package that can actually reach the granuloma. Again, it's location with regards to tuberculosis. If we can crack that, I think we got it, right? But it is tough. Those granulomas are built like bunkers, right? That is going to be huge if we can, as researchers can. I shouldn't include myself in there, but as researchers continue to improve methods of drug delivery, this is what I am hoping for at the very least. Okay, so that was the presentation. Are there any questions? Chat? Now we're in the black void. Let's go ahead and put the classroom back up. Any questions? This is an awesome paper. I think it's an awesome paper to cover. Not every paper, I think, has like knockout data that is like, oh, and then this is a beautiful like through line. Sometimes you get a paper and it's heavy and it's kind of complicated, and you have to like wrestle with it to figure out what you can and can't take away. Two-tail T for non-eliminated spleen. Still pretty good. Yeah. That makes sense. Thank you for doing the data analysis, by the way. Aerosolized test would be a next good step. Yeah. I think if you can aerosolize phage, that would be an awesome thing to try. I don't know if they have. They probably have. Like, you know, we can't be the first people to think of that, but I wonder what that progress has been like. They bred a new line of mice which better emulated human immune system. That almost seems more important. Yeah, I think the humanized mice is probably why this is a Nature paper and not so much the phage results. That's a good point, Spepik. Aerosolized tuberculosis, I think aerosolized phage. I think aerosolized tuberculosis would be ideal for research, but you probably can't do that safely. I don't know that you could do it safely. It would just be safer to not, though. So that might be an imperfection we just have to deal with. It illustrates how challenging TB research is. It is. TB research is tough. It is so hard. I wonder why it didn't work. Yeah, me too. I mean, my money is on it never made it to the lung properly. Injection through the bloodstream. I mean, there are small blood vessels in the lung, so I don't think that it is a pure function of just not being able to reach the lung because you injected through blood. Just because, right? Like, again, that granuloma is tough. I was doing a little happy dance because in the biochem lecture, I had hypothesized that phage would have difficulty reaching the granuloma, and this result, it's consistent with that idea. It's not supportive of that idea, it's consistent. It doesn't break the idea that phage are having trouble getting into the lung because of the granuloma. But then I read that actually phage can be endocytosed by mammalian cells, by eukaryotes, right? Which is interesting because phage are hyperadaptive to infect bacteria, but also our living cells can also do that. They can just absorb the phage as well, which I thought was really surprising. I don't know whether or not phage can phase through dead cells, which is an issue, right? Because the center of that granuloma are apoptotic macrophages that are dying or dead, and that is tough. They don't really do cellular processes at that point. At least I don't think they do, right? I've been surprised and proven wrong before with regards to the crazy, wonderful world of biology. Maybe a bit silly, but how did they choose which human immune cells to use for the mice? Was it one person's, homogenized, or what? Wonder if that has any meaningful effect on the results. That's a good question, 44. I think it was one donor per one mouse, and I think it was just like everything, because they used stem cells and then differentiated them into the proper immune cells. I don't know whether or not they added back all of the immune cells in these particular NSGs or just macrophages. It's a good question to look up or double check in the paper. Did they try the other phage? Maybe the lung is more agar-agar-like environment. I think they didn't because it looked more like drug resistance or phage resistance rather than the physical interaction. It's what is it? The simplest answer? That thing. Because while I think it is a possibility that maybe the phage act better on a physical medium when tuberculosis is growing on a physical medium, the simpler answer is that tuberculosis managed to get resistance because they were growing in liquid and because it's a much more friendly environment to split in. Yeah, listening to this, I think they were perhaps wrong in using mice. If they don't make granulomas, why not use pigs or apes or some animals that do? Our genetic tools in mice are unparalleled. We can engineer a mouse to do almost anything. It's insane. Like, there are experiments in mice that are so groundbreaking with regards to our application that we can't do in other animals because it's too expensive to develop those technologies in them. Right, guinea pigs are actually a pretty good tool for tuberculosis because they form granulomas. The issue with guinea pigs is that we don't have the genetic tools to do anything else. So if we set up this mouse, we can then start testing Like if we, okay, so imagine a future where we have this like quote unquote perfect humanized mouse has exactly our immune system, but in a mouse. Now we can start sorting with potential genes that are immune specific, right? So we can see if you have this gene in a macrophage, what does that do to tuberculosis? And we can't do that in a guinea pig or a pig or a monkey because our genetic tools have been made in mice. And now, why you might say, is there something intrinsic about a mice that makes it easier to have genetic tools? Yes, mice breed very quickly and they're very cheap to maintain. To practice and to refine a genetic tool for a pig, I think that's hundreds of millions of dollars. It would be insane to engineer a pig, because you would need so many. After about 10 minutes of Google, I am now a subject matter expert. Nice. Granuloma deformation is an active area of research with a couple of promising approaches. Very interesting. I am still not brave enough to do that on my own. Like if I were to relive my life as a graduate student, just because that problem seems so hard. Are there any cases of dead cells still performing some kind of active role? I don't know. That's why I checked myself, right? Like, but the definition of dead, but we know the definitions in biology are floaty, right? But the definition is that they are no longer doing cellular processes. But whether or not there are meaningful residual biologic activity, I'm not entirely sure. I await my Nobel Prize for posting this comment. Yeah, you'll have to wait about a year. Define dead. Yeah, hard to define dead, right? Everything in biology is hard to define. Occam's razor. Thank you, Mute. Thank you. Yeah, Occam's razor. What keeps a red blood cell alive? A functioning membrane and enough starting material. They don't need to make anything else. They are good the way they are. So they just start off with their material, and when they burn out and are no longer useful, they'll be recycled by the spleen. It seems like a giant mystery. How do the phages work? Why can TB become resistant to one but not to the other? Why do mice not form granulomas? Why do we? That's $1 million. Those are several $1 million questions, right? In terms of how phages work, I can actually explain a little bit of that, although my phage biology is quite rusty because the last time I worked meaningfully with phage was in graduate school. But phage work like any other virus, right? They inject their genetic payload, they make copies of themselves, and then they induce the bacteria to explode via lysis. It's lysis in bacteria because apoptosis, for the most part, is a eukaryotic thing. So there are lytic viruses that blow up bacteria, and there are lysogenic phages that incorporate stably in their genome or exist as episomes or something. Not episomes, but like actually, do phages form extra chromosomal DNA in bacteria? I actually don't know. But the point is, they blow up bacteria all the same. I have 100% successful method for elimination of TB in an animal host, unless of course host survival is a prerequisite. Yeah, exactly. I think someone else made that comment of like, it's not hard to find molecules that kill things. It's hard to find molecules that kill things and don't kill us. And that is very, very true. Strange idea, but would it be possible to design a bacterial expression system for these phages? There are some studies on protecting drug expression bacteria from the immune system for cancer drugs. Design a bacterial expression system for these phages. If Jim the Evo is around, he'd probably have a better idea than I do. My worry is that phages are pretty compact and don't really have a ton of room to stuff a bunch more things into. Right? They are pretty lightweight for the most part. So I don't know is what I will say to that. Why does animal immune system do X? Evolution. They're not human, and our last common ancestors were millions of years ago. Yeah, I know. I mean, that's always technically the answer, but I have a lack of a specific mechanism. I assume phages don't need to integrate their DNA into the chromosomes in bacteria. Yeah, there are lytic phages that don't need to do that and will happily make copies and blow up. Prokaryotes are pretty good at expressing random bits of DNA in the cytoplasm. Yeah, there's all sorts of extra chromosomal DNA. I don't know if phage are a contributor. Be interesting to look up. So what I can do is I can take a look at that paper and actually find out the answer to do the bacteria. Does the mouse immune system make antibodies against the phage? I know that they have the answer here. Just didn't put it up there. They did. Yeah, you can see it's figure 5. So it looks like there is an antibody response against the phage. And maybe that's why it's stuffing the response to the lung. Certainly possible, and certainly an unfortunate side effect of it being a foreign thing in the body, right? Yeah, so I know that there are a lot of people very excited about the potential for bacteriophage, and I am too, but it's complicated. Certainly very complicated. It might be like a desperation move, to harken back to Kingdom Hearts terminology, that you only pull out when antibiotics are not working. Do phage not affect eukaryotes because they only have RNA? Seems like they could mechanically attack eukaryotic cells. So the way that viruses in general work is that they will bind to a protein on the surface of a cell, and that will trigger the part of the membrane to, and be mature about this, chat, to invaginate, right? To collapse in on itself to form a little bubble to internalize that thing. So there are a couple of different pathways that this can happen. Phagocytosis is one of them, right? So when professional phagocytes like neutrophils and macrophages, when they go about eating things, they're using protein receptors that bind things that they need to eat. And then, when that protein binds, then you get that internalization. So they don't affect eukaryotes because they are evolved to bind bacterial protein and not our protein. And also they are evolved to take advantage of a bacterial DNA synthesis, RNA synthesis, protein synthesis machinery, and eukaryotes are just too different for phages to make use of our stuff. I would not be worried about a phage harming human health in that particular way. Yeah. Any other questions, chat? Or do we want to hypothesize more about how to tackle tuberculosis? Why haven't they really evolved for ourselves? Well, why would they? There are bacteria everywhere. It's like asking, why eat a McDonald's hamburger? Why pay for a McDonald's hamburger when you can go to an all-you-can-eat buffet for free, right? Like, some entities might eventually evolve to prefer a single McDonald's hamburger, but a lot of entities would probably stay in that all-you-can-eat buffet, right? As a neutrophil, since you mentioned phagocytosis, what is the best tasting bacteria to eat? Here's the thing. As I have been building out the videos for my channel and learning a lot more about immunology, a lot of them really suck. A lot of the pathogens that we care about, like gonorrhea, just like blow up neutrophils from the inside. So I'm not feeling too good about eating any of the bacteria at this point. It's kind of crazy. I mean, it's survival bias, right? Neutrophils are great at killing the bacteria that don't hurt you, but the bacteria that evolved to hurt you likely evolved a way to hurt neutrophils too. So I find myself, when I'm researching all of these different pathogens, that I find a lot of them do terrible things to neutrophils, so that makes me feel very bad. Bacterium food review. 1 million subscribers. Let's do it. 1 million subscribers. I'll review every bacteria as a food treat, because we all know that food tier lists are very, very popular. How common is TB outbreaks in non-human worlds? I actually don't know what the animal reservoirs of TB are. I believe that there are non-human reservoirs for TB, but I think TB is at least most devastating in humans, although I would need a fact check on that. What's the mechanism? Can you remind me what mechanism you were talking about? Based on your lore video, I would have thought you haven't eaten a bacteria before. Yeah, well, I am one. So yeah, the lore video is that I am a lover and not a fighter. I am a neutrophil with deficient properties. I am maybe perhaps conveying the sentiment of my neutrophil brethren. But yeah, if you go watch my lore video, which I still occasionally go back to watch, I think I did a pretty good job on that. I'm an experimental neutrophil, intended to be a great bacteria hunter, but I just broke everything. And instead of being recycled and incinerated, I was made to stream and make YouTube content. If I did eat the SCOBY though, that is true. I did eat the SCOBY. So a SCOBY is the gelatinous disc of bacteria and yeast that forms on top of your kombucha. And that was actually not bad at all. That tasted pretty good. That lore video is peak. Thank you so much. Neutrophil VTuber, is that a kind of furry? Technically, I'm a part of a human. I don't think that counts as furry. I'm technically just a subsection of humanity, you know? Would you call like a nose VTuber a furry? I don't think so. I don't think so. I want to chat a little bit about how the classes have been going, just because I think there are enough biology people or students in general. It doesn't have to be students. I think there are enough people in here to pitch the idea of, like, you know, my students right now are pitching their ideas to tackle all sorts of different novel problems. I'm not going to tell you which problems that they're tackling, because maybe they'll become really good ideas one day that they'll want a patent. But they're also thinking about novel ways to tackle horrible diseases and stuff like that. And I was wondering if chat would be interested in submitting your ideas to solve terrible problems using biology and have me review them. I don't know. I think it'd be a lot of fun, especially for people who aren't into biology, to have your idea refined is what the teaching language would be, right? Not torn apart or roasted, but refined. Because I really like, especially because, like, some of you have had ideas that I either ignored, and I'm specifically referring to CR, who had brought up the antibody response thing, and Pamela, you are not biologists, but you thought of something at the level of a professional biologist, which I'm so, it's unfair to say proud, because I don't think that I deserve credit for that, but I am proud of having the space where that could have happened, right? Because the fact that you out of pocket predicted something, and not only predicted something, but predicted something that I was not going to show, that was away from my story, no foreshadowing, you know, without my help, you were able to come up with something at the tier of a professional biologist. It just makes me so happy. So I would really love to probe your brains for all sorts of other creative approaches and ideas you have for biology. I think that'd be really cool. Driving by to say hi and mention a fun fact: Pseudomonas aeruginosa smell like a wet tortilla. Oh, interesting. Would not want to ingest them, although as long as you have a functioning microbiome, it's probably not that bad. Is there anything similar enough to TB that we can get some ideas for treatment from? Other mycobacterium, which are used as substitutes for TB because it's too dangerous to work with TB a lot of the time. Pseudomonas smells like grape cough syrup to me. Oh, interesting. Yeah, I was a fake microbiologist, so the only bacteria I've ever worked with is E. coli, unfortunately. I'm sure that there are a lot of other cool bacteria to work with. I hope you just make your students watch your YouTube channel, since I imagine we're effectively watching your lectures. Not really. My lectures are a lot shorter. So I teach a lab class. So I teach in an environment where they are actively encouraged to think. But they are thinking about the science that they are doing with their own hands. So it's a little different. We do a journal club assignment, so that is pretty similar. So the presentations that I would give, they would give, and I would grade them. But I will never tell my students what I do, because I talk about them. What's the point of being a teacher on the internet if you can't talk about your students, right? But I would hate for them to, like, find out, oh, it was like, not that I would ever say anything bad about them, but, like, you know, you never know, right? So effectively, they will never know. And even if they go like, "Hey, are you Phage the neutrophil?" I'll never say yes. I will deny. Could be cool, but you could maybe list some of the biggest problems that need to be solved. I would also be curious as to what they would be. I mean, that would be my own bias, right? Like, in order to get a really good idea, you would have to poll so many different people. But I can do a tier list of problems that I think are really pressing, or maybe, oh, that's an idea. I could do a tier list of my most favorite biology problems to think about, and that would be cool. That'd be very cool. The closest I've been to being a biologist is when I got swine flu one time. Flu is something that I will definitely make a video about. It is very important. If we are talking about biggest problems to solve, the flu, that is going to probably be in my top 10 in terms of biology problems. Here's my crackpot idea. What if we're going at this backwards? Maybe we should try to make more granulomas to protect the body from the virus. If I recall, most TB infected are asymptomatic because of these. It's certainly something that people have played around, not inducing more granulomas because that's technically damaged lung tissue, but rather this idea that, so there was this dominant idea that the granuloma was like, not dominant, maybe that's not fair, but there was this idea that the granuloma was evolved to protect the body, but now that's become a lot more nuanced as people are now thinking that TB is taking advantage of it. So it's a push and pull evolution stalemate, basically. I take your class and blackmail you for an easy A. Again, I would never even confirm. I am blackmail free. Even if you tell people, I'll still never confirm. Five playing the ultimate Among Us. That's true. That's true. There's loads of different flu viruses. Yes, very, very, very many flu viruses. Well, a lot of influenza subtypes. We'll see. Aren't large granuloma loads mechanically damaging? I could not say that with factual, like, for sure, but it is technically damaged tissue, so I would assume that it hurts lung function. Why are colds common? Colds I will do at some point in the future. I just think that let's just say, given the current political situation in my in the country that I love so much, the great US of A, it might be more important for me to talk about the flu first versus the cold, although the cold is definitely on the list of things to talk about. All right, so it is getting late and Purkinje needs to be tube fed through her esophagus. It is that time. I will make a plug for the Discord. So let me go ahead and put that link up there. If you all want to continue the conversation, I'm pretty active in the Discord. I'm quite the active chatter. So, you know, don't be afraid to pop in. And what I will say again is please go ahead and check out Myelin and Syzygy, the two newest VTubers to join us in VTHEOREM. Myelin doing psychology and Syzygy doing physics and astrophysics, engineering, and CS. They are very fun people and I look forward to working with them in the future. And I look forward to talking to you all at some point. A very sad story that I'll end on is we were supposed to go to the beach and sip margaritas on the beach where it's warm and be wined and dined. We had this lovely vacation planned, and because Purkinje's health is not the best, we've had to cancel that. So while I am quite sad about that, that does mean that I might stream again this week and not take Thanksgiving off because I love streaming and I love making content for all of you. And I think in line with this tradition that I've just recently decided, it might be a just chatting stream where I wear like a, I don't know, pilgrim hat or something. So, you know, maybe look forward to that in the future. So I will see you all later. I hope you have a wonderful vacation if you are celebrating Thanksgiving. And yeah, goodbye.